discussion of iron metabolism today.
Is it good?
Before I start with today's discussion, I wanted to address some of the questions regarding the stoichiometry of iron, transferrin, and the transferrin receptor that came up last time. And I'm not going to ask stoichiometry questions on the test, like how many irons, how many transferrins, how many receptors. What you should know is that ferric iron is bound by transferrin, and then the transferrin is bound by the transferrin receptor, and that's endocytosed. But I did want to address this question about stoichiometry, because it's a good question that came up last week on Wednesday. So I did some digging into the literature, and in the mid to late 90s, they determined the crystal structures of transferrin and the transferrin receptor. So on the left, we see transferrin. This consists of an N and C terminal lobe that are highly similar to each other, and these are joined by this helical linker. But this is one transferrin protein, and each of these lobes coordinates a single ferric iron atom. So we see in the core of this is the ferric iron. And so one transferrin combined two ferric iron atoms, one on each lobe. And then this is the crystal structure of the dimeric transferrin receptor. So we see the lobes, and it has this really nice looking sort of butterfly structure. And so the question after these were determined, the structures were determined separately, was how these might fit together and how the receptor might recognize transferrin. And this would have consequences for the stoichiometry of these interactions. And so there have been different models that came out just following these structures. One idea was that this transferrin could just sit right into this pocket or this cup formed by this butterfly-like structure, right? And then another paper modeled a binding interface on the side of transferrin. And this would suggest that maybe it could bind two transferrins. So these are very different models, one binding to the side or one binding to this cup, and one have very different consequences on the amount of transferrin that could be bound by these receptors. So the answer came from Tom Waltz's group at Harvard, who determined the structure of the receptor together with transferrin. And this was a cryo-EM structure. And this showed the transferrin receptor bound to two transferrin molecules, two transferrin proteins. And this was actually a really different structure than expected. So neither of the predicted models in the past predicted this structure. Because you can actually see here, this is a receptor, and the transferrin, the N and C lobes, are actually kind of binding to the bottom of this receptor, not to the top. But this unequivocally demonstrated that the transferrin receptor could bind two of these transferrins, each bound to two irons. And this was confirmed by a crystal structure showing the same thing, that you have this transferrin receptor shown here in the center, and it could bind two transferrins here, the N and C lobe on one side and the N and C lobe of another transferrin on the other side. So this means that each of those dimeric transferrin receptors can bind two transferrins, which each bind two ferric irons. So this was very nicely represented in an illustration aptly titled Mysteries of the Transferrin-Transferrin Receptor 1 Interaction Uncovered. And so this shows the dimer of transferrin receptor, and it's binding to two transferrins, each with two irons attached. These are released to DMT1 and ferroreductase for transport out of that endosome, and then the transferrin receptor and transferrin can be recycled back to the cell surface. So if we return to this diagram that I presented last week, this appears to be correct, that these transferrins each have an N and C terminal lobe, each bound to ferric iron, so two irons. And then the dimeric transferrin receptor combined two transferrins, so four irons. Now, I'm not going to get really specific with the numbers of iron in terms of the tests. You should know that transferrin binds ferric iron, and then this is recognized by the transferrin receptor. But I just wanted to come back to this since this question came up last time. So before I go on, at the beginning of the semester, I was doing these polls with kind of example questions. And I just wanted to get a sort of raising of hands if people found this useful. Usually these were questions sort of reviewing the last lecture and were kind of practice questions. So maybe raise your hand if you found this useful. Pretty good. And not useful? Okay. The useful has it. So I'll continue to do this just to give some example questions. It's also useful for me to get a sense of how well you guys understand the material. And so again, you can respond by going to this website, tolev.com/NST103, and it'll give you the question and the answers and the choices, or you can do this by text message. So you text NST103 to this number, and then you can answer by texting these various letters, right? Just like we did at the beginning of the semester. So this question has to do with absorption. So iron is absorbed at the enterocyte apical membrane, so from the gut, in blank form. So ferric or ferrous iron via which transporter? The divalent metal transporter, DMT1, or ferroportin? I'll give you guys a second to kind of log in there and try this out. How you guys are doing? Okay, so we have a mix and a readjustment of the answers. So it is in fact ferrous iron via DMT1. So if we just recall, this was kind of the overall diagram of iron absorption. And we had this transformation of iron between the ferrous and ferric forms in order to cross the membranes and for storage. This was kind of reminiscent of like triacylglycerides, right? That had been broken down, and then remade into triacylglycerides, and retransported out, and then rebroken down to be taken up into adipocytes. So iron is similar in that in order to be taken up, it has to be transformed into ferrous iron. So this can be present in our diet, or it's converted by these cytochrome B, the duodenal cytochrome B, from ferric to ferrous. This is taken up by DMT1. This can either be stored or transported out through ferroportin, where it's converted back to ferric iron via hephaestin or ceruloplasmin and packed onto transferrin for transport. Okay. So from the apical side, it's taken up as ferrous by DMT1, and the basolateral side, it's transported out by ferroportin, also in the ferrous state. Okay, let's try another one. So this one has to do with the IRE. So binding of iron regulatory protein to a 5' iron response element, or IRE, will have what effect? So decrease protein degradation, decrease transcript stability, decrease gene expression, or decrease protein translation. The 5'. So you can have 5' or 3' IREs. So it's pretty good. The majority has it in protein translation. And now almost everybody got it. That's good. So just recall that these iron regulatory proteins, they're really cool proteins because they allow the cell to directly sense iron concentrations. And it does that because it has this iron sulfur cluster. So under conditions where we have sufficient amounts of iron, it'll have this cluster and it forms this closed conformation. If there's not enough iron, we no longer have this full cluster. It adopts this open conformation. It combines the IRE. And we can have these IREs, these iron regulatory elements, at the 5' or the 3' end of the transcript. So remember, this is on the mRNA. A lot of times we talk about gene expression and transcription. This is actually on the mRNA, the transcript. So at the 5' end, this will decrease protein translation. You can think of this as like the ribosome is going to bind at the 5' end and track along this transcript. We've got this giant stem loop structure with a protein bound to it. It'll probably disrupt ribosome tracking along this and decrease translation. And this is going to be important for things like ferritin, ferroportin, and aminolevulinic synthase, ALA synthase, which we'll talk about today, and heme synthesis. If we had a 3' end, this is involved in regulation of transcript stability. So this might not affect the tracking of a ribosome along it, but it affects the stability of the transcript. And this is going to be important for transferrin receptor. one more. So this is also with reference to regulation of iron, this time plasma iron. So hepcidin regulates plasma iron concentration by decreasing uptake through DMT1, increasing iron storage in liver ferritin, decreasing oxidation of iron by hephaestin, or decreasing ferroportin levels by inducing its degradation. Okay, so the majority says this, and this is correct. So it decreases ferroportin levels by inducing its degradation. So remember that hepcidin is the regulatory factor released by the liver, so it's going to be in the plasma, and it's dealing with the concentration of iron in the plasma. And it does this by binding directly to ferroportin. So it directly affects the transporter on the basolateral side of the enterocyte that transports iron out into the plasma. So it binds to ferroportin and induces its endocytosis and lysosomal degradation. So if you have high iron in the plasma, you're going to have high hepcidin release. It's going to bind to ferroportin, downregulate that transporter, and decrease iron transport into the plasma. If you have low iron in the plasma, you have low hepcidin, you have increased transporter in the plasma. Okay, so we're going to move on from here to talk about heme and red blood cells today. So we'll talk both about the structure and the synthesis of heme, as well as its degradation, and then its importance in erythrocytes and connections with nutrition, iron deficiency, and anemia. So in our last lecture, we discussed various aspects of iron metabolism, some of its useful properties in chemistry, how it's absorbed, how it's distributed through the body. And some of the most important aspects of iron in our body revolve around its incorporation into heme. So this is a very characteristic chemical structure, and it's utilized very widely as a protein prosthetic group. This includes things involved in oxygen binding, so hemoglobin. So this is the four globin subunits, each coordinating a heme, which each can bind oxygen through that iron, as well as myoglobin, which is a single subunit coordinating a single heme, which combines single oxygen. But it also can be used in enzymes involved in oxygen metabolism, such as peroxidase and catalases, electron transfer, such as the cytochrome proteins and detoxification reactions, or electron transport. And so all of our cells have the ability to synthesize heme. There are a lot of proteins that need heme in order to function. However, there are some cells that can't. So there are a few exceptions, such as mature erythrocytes. But the precursor cells to erythrocytes synthesize a ton of heme in the presence of hemoglobin and really pack it into these mature erythrocytes. We'll talk about these cells and why they're unique towards the end of the lecture. So this is, the heme is referred to as a porphyrin structure. It has this porphyrin ring. And porphyrin is the simplest porphyrin. And it has a few different features. It's considered a heterocyclic organic compound, so it has carbons as well as nitrogen, so hetero. And it has these 4 ring structures. And these are referred to as pyrrole rings, so it has 4 pyrrole rings. These are 5-membered rings containing nitrogen. And each of these 4 pyrrole rings are connected by these methane bridges, right? So we form a tetropyrrole ring. Okay, so we have 4 pyrrole rings connected by these little methane bridges. Okay? And when we look at heme, we can see this very similar structure to this porphyrin. We see again these 4 pyrrole rings containing these nitrogens, as well as these methane bridges joining these all together. Right? And then we see some unique things about heme relative to porphin. Of course, we have the coordinated iron in the center, coordinated by these nitrogens. And it has 2 additional binding sites, which could be used to bind oxygen or a histidine within a protein. And then we see a variety of added side chains. So there are 4 methyl groups, 2 vinyl groups, and 2 propionic groups. And this is what makes heme heme. And I just wanted to point out, this slide is just one I added this morning, that this structure, this porphyrin structure, isn't just unique to heme. It's a really, really ancient chemical scaffold that's widely used throughout evolution. So we can see things like chlorophyll in plants using a very similar porphyrin ring, coordinating magnesium. And this is vitamin B12, which we'll discuss on Wednesday, also known as cobalamin, using a porphyrin ring to coordinate cobalt. Okay, so this type of porphyrin ring structure is really prevalent throughout evolution and prevalent throughout various organisms to coordinate things like cobalt, magnesium, or iron, and utilize this for different enzymatic reactions. Okay, so we're going to walk through some of the main steps of heme synthesis. We won't go through all of the reactions, and I'll try to stress what is important to know. In the synthesis of heme, there are 8 separate chemical reactions. What's interesting is this is distributed between multiple cellular compartments. 4 occur in the mitochondria, and then 4 occur out in the cytoplasm. Okay, and this first reaction is mediated by levulinic acid synthase. And whenever you hear synthase, it refers to enzymes that's like joining 2 things together. So it's pulling 2 things together. And indeed, that's what ALA synthase does. It pulls glycine and succinyl-CoA together. So we have glycine, our amino acid, succinyl-CoA, an intermediate in the TCA cycle. And it's taking these 2 molecules and synthesizing 5-aminolevulinic acid, also referred to as delta levulinic acid. This is levulinic acid here, and it has this this amine group off of the 5th carbon. So this ALA synthase performs the first rate-limiting step and the committed step in heme synthesis. So not surprisingly, this is going to be a highly regulated step, and I'll tell you about its regulation. In addition, it requires pyridoxal phosphate, or PLP, which is also known as vitamin B6, as a cofactor. Okay? And this is all occurring in the mitochondria. So ALA synthase, aminolevulinic acid synthase, forming aminolevulinic acid from glycine and succinyl-CoA. Okay? And so, not surprisingly, since this is the rate-limiting enzyme in the committed step in heme synthesis, this is an important step of regulation. And I already referred to one point of regulation for this enzyme, and that's through a 5' iron response element, so an IRE. So in addition to ferritin, which stores iron, or ferroportin, transports iron out, ALA synthase is controlled by a 5' IRE. So under conditions of low iron levels, the IRP will bind this 5' iron response element in the transcript and decrease translation. So low iron, you'll get low amounts of this enzyme, and you'll get lower production of heme because there's not enough iron. Right? That makes sense. In addition, this is subject to really classical kind of product negative feedback regulation. So at the end of this pathway, we produce heme, and heme is going to feed back and downregulate synthesis of ALA synthase. So it decreases the transcription of ALA synthase, so it decreases the levels of ALA synthase, and it also blocks the translocation of ALA synthase into the mitochondria, which is a site of action where it's joining that glycine and succinyl-CoA. And so both of these will decrease overall flux through the heme synthesis pathway. So if you have low iron, you're going to not want to be synthesizing heme. If you have a ton of heme, you also have negative feedback regulation of this pathway. Okay. So we have this 5-aminolevulinic acid. This can be joined together to form our pyrrole ring, porphobilinogen, and this occurs through ALA dehydratase. So it's joining 2 ALAs, 2-aminolevulinic acids, to form this chiral ring structure. And this is occurring in the cytoplasm, and it requires zinc. And it's also interesting that this enzyme is inhibited by lead. So it's going to be important in lead poisoning, which can lead to anemia. And there are actually two enzymes, this ALA dehydratase, as well as a second enzyme in the pathway that are both inhibited by lead, which will influence lead-induced anemia. Okay, this porphoglinogen, which has this chiral ring, can then start to be linked together to form our heme. This occurs through the actions of porphoglinogen deaminase, so it's removing this amine group and linking these porphoglinogens together, linking the chiral rings, and we're starting to see what looks like the porphyrin ring structure of heme emerging now, right? So we have this porphoglinogen deaminase, it forms hydroxyl methylbilane. This is all occurring in the cytosol. We now have a tetrapyrrole molecule, but it's not joined quite yet. Okay? So we've joined together these chiral rings, tetrapyrrole molecule, and finally this uroporphyrinogen 3, also in the cytoplasm, can link these together, and we have a completed porphyrin ring. Right? This again, it's occurring in the cytosol. Okay, and so there are a variety of different reactions that occur that modify this structure. I'm not going to go through all the details of those reactions, which I'm sure you're probably happy about. And this will form the protoporphyrin 9 structure. And this has all the, mostly it just is modifying these side chains. So it gives rise to these four methyl groups, and the 2 vinyl groups, and the 2 propionic groups. So just a series of reactions modifying the side chains. So we already have our porphyrin ring. And then we have, this moves back into the mitochondria, and we have the final enzyme in heme synthesis, ferrochelatase, which adds the iron to the center of our porphyrin ring, okay, and giving us finally our final heme. And this enzyme also can be inhibited by lead. So those 2 enzymes, the ALA dehydratase and this ferrochelatase, can be inhibited by lead, and this is important for lead poisoning and the anemia associated with lead poisoning. So this is the full reaction pathway. I didn't talk about all of these steps, and I tried to kind of stress the important ones. The most important one I want you to remember is this first one, the ALA synthase reaction, since this is the rate-limiting step, the committed step, and the most highly regulated. So you should try to know the mechanisms by which this is regulated and the fact that it requires that vitamin B6, the PLP, as a cofactor. The other 2 steps that I'd like you to remember are the dehydratase step and the ferrochelatase step, since both of these can be inhibited by lead, and they're important for lead-induced anemia. So that's the dehydratase and the ferrochelatase steps, right? And this I kind of like this pathway map just because it kind of shows, it's easy to appreciate that this is occurring in these different subcellular compartments. So you can see that this first step is in the mitochondria, these intermediate steps are taking place in the cytoplasm, and then these final steps again present in the mitochondria. So it is this dynamic kind of movement of these metabolites between the 2 compartments, which is interesting. Okay, so kind of major steps is a lot to take in there. So I'm just going to go through some of the major steps and key points that I'd like you to remember. So first off, the heme synthesis is occurring in all cells because there are a lot of different types of proteins and enzymes. They utilize heme as a prosthetic group in their chemistry and reactions. There's a particularly high amount of heme synthesis happening in the bone marrow, giving rise to the hemoglobin and red blood cells. So that's not surprising because when we talked about where all our iron is in our body, remember 2.5 grams of it is present in our erythrocytes, in our red blood cells. That's all being packaged in the marrow. So the bone marrow and the cells giving rise to these red blood cells really amp up their heme production and to form the hemoglobin for red blood cells. There's also quite a bit of heme synthesis happening in the liver to give rise to the cytochromes, which are important for detoxification. some toxic molecules entering our body. Okay, so I want you to remember the ALA synthase. This combines glycine and succinyl-CoA to form that aminolevulinic acid. It's happening in the mitochondria, requires PLP or Z6 as a cofactor, and it's an important point of regulation, both with the 5' IRE and as well as by negative feedback by heme, right? The product of the pathway. We talked about the dehydratase, which is susceptible to inhibition of lead. This gives rise to the porphobilinogen, that first pyrrole molecule. There are then a series of reactions to connect these porphobilinogens to form that tetrapyrrole and to form the ring. This is modified and moves back into the mitochondria. And then remember this enzyme, ferrochelatase, which mediates the final addition of iron to the core of the heme and also can be inhibited by lead. So we have two enzymes inhibited by lead. Okay, so heme also has to be degraded, and this is also going to be taking place in a lot of different cells. So when we talked about iron absorption from our gut, remember it can be taken up as ferrous iron or as heme. If it's taken up as heme, it's first, the iron is released by cleaving that heme ring, and that iron can then be secreted through ferroportin to the body. In addition, so it's happening in enterocytes when we take up dietary heme, and then the other site, probably the main site, is in the spleen and macrophages as they break down old red blood cells. It's going to be a ton of hemoglobin and a lot of heme degradation and recycling of that iron. Okay, so we have to degrade heme to recycle the iron that's stored inside the heme, right? And the first enzyme involved in the breakdown of heme is heme oxygenase. So this cleaves the heme ring to form biliverdin, and it releases the iron from the center of the heme. Okay, this molecule, biliverdin, then undergoes partial reduction to bilirubin, and this is mediated by an enzyme called biliverdin reductase. This is all happening in the same cell. This molecule, bilirubin reductase, isn't metabolized further in these cells. It has to be transported to the liver. So bilirubin is released into the blood and complexed with the protein albumin and transported to the liver for further metabolism and excretion. We kind of have this inter-organelle relationship going on with the breakdown of heme. Okay, so just a reminder, heme is being broken down by heme oxygenase to biliverdin, then to bilirubin. This is released into the blood, transported to the liver, where it's then modified by this enzyme with this really catchy name, UDP glucuranosyl transferase. It adds glucuronic acid to the propionic side chain of the bilirubin molecule to form this bilirubin diglucuronide. Okay. And this is important because this molecule is a molecule that can be secreted by the liver into the bile, which is then secreted into our small intestines for excretion from our body. Okay, so this is a route to get rid of this molecule. Okay, so that's why it's important to be able to form this bilirubin diglucuronide. Interestingly, these different breakdown products have different colors. This is kind of red, this heme. Biliverdin is more like green, and this bilirubin is yellow. And that's what gives rise to like bruise, the changing color of your bruise. When you first get a bruise, it might look more purple, but then as it turns kind of yellowish. This is all related to the breakdown of the heme into bilirubin and biliverdin and bilirubin, which have different colors. Okay, so this is just a representation of that full pathway which we just discussed of heme degradation. And this is kind of a nice representation of the pathway because it gives indicates the different tissues this is happening in. So, for example, this is showing the most prevalent amount of heme degradation, which happens from aged erythrocytes. But this will also happen to some degree in other cell types as they degrade heme-containing proteins, or in your enterocytes as you get dietary heme from meat in your diet. But the main amount is coming from aged erythrocytes. This is happening in macrophages in the spleen. So you have hemoglobin from those aged erythrocytes. This protein is degraded. The globin is degraded all the way down to amino acids for recycling, and that heme is released. And then the heme is further degraded so you can recycle the iron. So heme, the ring is opened by heme oxygenase, giving rise to that biliverdin molecule. And these different MV, MPs just refer to the side chains, the methyl, vinyl, and propionic side chains. And then that biliverdin is partially reduced to bilirubin. This is what can actually be transported. So the bilirubin can then be transported in the blood in conjugation with albumin to our liver for the final metabolism. So the conversion to bilirubin diglucuronide, which can be excreted in our bile. Right? Does that make sense? Okay, let's take a 5-minute break and then we'll come back with red blood cells. Some of its structural features, how it's synthesized, and how it's degraded. One of the most important roles for heme in our body and the main site of the majority of our iron in the body is going to be in hemoglobin and in erythrocytes. We'll spend a little bit of time talking about red blood cells. So when we refer to hematopoiesis, this refers to the production or development of blood cells. So poiesis is production or development, and hemato is referring to blood. And in our blood, we have a variety of different cell types. We're specifically going to talk about erythrocytes, which can contain high amounts of hemoglobin and high amounts of heme. And so erythropoiesis refers to the development, differentiation, and maturation of erythrocytes or red blood cells. And these terms can be used interchangeably, red blood cells or erythrocytes, these red cell types. Okay, so erythrocytes, if we just look at the numbers, it's pretty impressive when we think about it. So there are 2.4 million new erythrocytes produced per second. That's pretty amazing. So in our body, we have an incredible amount of resources going towards the production of red blood cells. And there are a lot of them. So approximately a quarter of the cells in our body are red blood cells. There are a ton of red blood cells circulating through our body, and we're putting a lot of effort into their production. And within these red blood cells, within each red blood cell, they contain 270 million hemoglobin molecules. It's a crap ton, right? So combined, within each red blood cell, each of those hemoglobins bind 4 oxygens. So these cell types are incredibly evolved for the movement and transport of oxygen, right? Through the production of this much hemoglobin and this much capacity to bind and move oxygen through our body. Of interest, in 2008, it was reported that they had successfully taken human embryonic stem cells and coaxed these into becoming erythrocytes in vitro. And it's hoped that these types of artificial erythrocytes could eventually be used for blood transfusions, which would be pretty amazing for medicine. So there's a lot of effort to get people to donate blood, but there are a lot of problems with this, right? Sometimes we have to store that blood, the blood's of different ages. If you can produce it in vitro, you can take someone's adult stem cells, convert them into erythrocytes, and produce brand new red blood cells without any bloodborne pathogens. It's genetically identical. So there are a lot of advantages about this. And I think some of this has entered clinical trials maybe this year, next year. But whether or not it's actually financially possible, I'd be curious to know the kind of money it takes to produce therapeutic clinical amounts of erythrocytes in vitro versus just getting them donated by another person, right? So those two things, whether or not it will be financially viable, is definitely still up in the air, but it seems to be possible at this point with what we know about erythrocyte differentiation. So these, as I mentioned, are really unique cells. Just this one slide. Let me try exiting. Okay, hold your breath. Okay, so these are really unique cells. They're very different from most of the cells in our body, and they're highly specialized for their task. And so this is something we've come across as we've talked about cell types in other tissues as well, that each cell type within our different organs are highly specialized for their function. So enterocytes, for example, have villi, and gastric cells secrete things to protect them from the acid, right? All these different cell types are highly specialized. But most of the time when we think of a cell, we think of it being packed full of organelles, so it has a nucleus, has ER, it has mitochondria, a variety of organelles. But red blood cells, mature red blood cells, are really unique in that they lack a nucleus and they lack organelles, which seems really weird, right? And they also have this very unique shape because of the lack of the nucleus and the organelles. They adopt this biconcave shape, kind of like a little biconcave disc. And this is an important shape because it maximizes the surface to volume ratio of the cell, which is really useful in terms of exchanging gases. So it maximizes the amount of surface area available for exchange of oxygen. Okay. This shape is also really useful. It's a small shape and it's very resistant to the stresses of being pushed and pulled through these very narrow capillaries, which has a lot of shearing stress and collisional stress that maybe other cell types might not have to be exposed to. So this shape is useful for that. And these cells live for quite a while in the blood. They're around 120 days lifetime, and at which point they might either rupture or they're actively degraded by the body. So the body recognizes these aged red blood cells, actively degrades these, and then recycles the iron into new red blood cells. So there's constant degradation, release of that iron, constant resynthesis and incorporation of that iron into new blood cells. And this all happens, this erythropoiesis, the development of mature red blood cells, happens in the bone marrow, which is found in the center of most bones. And if you think about bones, they're not actually completely dense. They have this porous structure. There's a lot of blood vessels that run through bones, and within this it also contains bone marrow. And this is where the stem cells that produce our red blood cells are located. And this will produce a variety of different blood cells, including our erythrocytes or red blood cells. Okay, and so here is just a diagram or an illustration of the maturation pathway of an erythrocyte. So this is showing the hemocytoblast, also referred to as the hematopoietic stem cell. So this again is residing in our bone marrow and can give rise to a variety of blood cells. So in response to a signal, it's going to divide, and one of these cells will stay a stem cell. The other can be committed to an erythrocyte differentiation program. And so the first stage of this is the pro-erythroblast, and this is a committed step towards the production of this mature red blood cell. And so over time, there are going to be a variety of changes, including a decrease in the size of the cell, a decrease in condensing of the nucleus, and a loss of mitotic activity. There's a lot that happens as this differentiates into a red blood cell. So if we just kind of break this down into a few different phases of development, in the first phase, there's actually an expansion of the number of ribosomes. So it starts to produce a bunch of ribosomes because it's going to want to produce a whole bunch of hemoglobin. And so first expands its pool of ribosomes. Of course, they're involved in protein translation. So this is the early erythroblast. As we move into the late erythroblast and the normoblast stages, we're going to have extensive continued production of hemoglobin, and this is going to become really dense. Remember, it has hundreds of millions of hemoglobin protein molecules packed into this cytoplasm of this tiny cell. And what's really fascinating about this end stage of maturation is this loss of organelles. And this is really dramatic. So it takes this nucleus, over time this nucleus, the DNA is becoming highly, highly condensed and packed in to this smaller nucleus, and then it extrudes the nucleus. It actually pushes it out of the cell, which is really fascinating. And in the final stages, it loses the rest of its organelles. These can either be degraded by autophagy, which would mediate the transfer of some of those organelles to the lysosome for hydrolysis and degradation in the lysosome, or those organelles can also be extruded out of the cell. So it's really neat to think about what kind of cell biology would be taking place to push a nucleus out of the cell. And so this gives rise to this mature erythrocyte, which has this unique shape, lacks pretty much all organelles, and is highly packed with hemoglobin. And so that's why you have no ability in this mature erythrocyte to make hemoglobin. You don't even have mitochondria. Right? That's why they rely so heavily on glycolysis, because they don't have mitochondria at this stage. They can't undergo cell division or any regulated gene expression, because they don't even have DNA. Right? So these are kind of at a very terminal cell stage, and they move around in our body for about 100 days, and then they're degraded and we make humans. So at this, we have this ejection of the nucleus and ejection of other organelles. Okay, so this step of extrusion of the nucleus is referred to as enucleation, and I think it's a really neat kind of morphological change, and it's really dramatic. And these are microscopy images of unstained cells or stained cells. What you see is that there's this slight protrusion where this arrow is as the cell actively starts to push its nucleus to the edge of this cell. You can see this bulge forming. And here you can see it really nicely, this bulge forming from the cell. And it's actually very much like mitosis, where you get that cytokinetic furrow when you have cell division, except here it's pushing the nucleus out and it forms this cytokinetic furrow, and then this will slowly pinch off and the nucleus will be separated and degraded by surrounding macrophages. And then we'll leave a cell that's just basically a bag of cytoplasm and hemoglobin without a nucleus. And you can see that this dramatic cell biology is really fast. These are in minutes. This is in 3.5 minutes. It's pushing that nucleus out, forming the cytokinetic furrow, and separating that from the cell. And so you can also see that here very nicely in these stained cells. The early erythroblast, moving to the late erythroblast, that nucleus is becoming highly condensed. And then it's pushed out, and you have this contractile ring, just like mitosis, and it's separated, giving rise to the reticulocyte, and then finally our red blood cell, which is really packed with hemoglobin. Okay. So this, not surprisingly, is subject to an amount of regulation. One of the most important factors is this protein erythropoietin. So it's a small glycoprotein, so it's glycosylated, and it's secreted from the kidneys. Okay. This would be going out through the endoplasmic reticulum, through vesicles, and then exocytosed into circulation. And specifically, this is in response to low levels of oxygen. So the kidney is sensing levels of oxygen in the blood. There are low levels of oxygen. It increases the production and the synthesis and secretion of erythropoietin. And then this circulates through our blood and binds to erythropoietin receptors on our erythroid progenitor cells, those hematopoietic stem cells. And it signals that the body needs more erythrocytes, and stimulates differentiation of these stem cells into our red blood cells. So this increases the erythrocyte production, which enters the blood, and increases oxygen delivery. So in this case, we have an inverse relationship between erythropoietin levels and oxygen. So low oxygen levels, we up the erythropoietin. We have normal oxygen levels, erythropoietin goes down, and somehow we find this nice balance of producing red blood cells and maintaining our oxygenation of our tissues. Right? So the erythrocyte breakdown, so it's produced in the bone marrow from these hematopoietic stem cells, circulates in the body for about 120 days, and then it comes back and is degraded by macrophages. This is mostly going on in the spleen, but also is happening in the liver and in the bone marrow. In these macrophages, this is an actual transmission electron microscope image that's been false colored, and you can see these macrophages eating the phagocytosis of red blood cells. So it's eating up 3 different red blood cells, kind of engulfing them for degradation. And these are relatively, you can see how small the red blood cells are relative to that macrophage. It's able to engulf this and mediate its degradation. So here's a diagram of this macrophage cell. The red blood cell is being phagocytosed and engulfed by the macrophage. It's being degraded in the phagolysosome, so that phagocytosed bunch of lysosomes merge with this structure, and the enzymes within the lysosome can mediate degradation of all of the red blood cell protein and lipid components. And so this is going to release globin, which can be then degraded to free amino acids, and also our heme. And we've already been through heme degradation, right? So heme oxygenase will come in, release iron. This iron can either be stored as ferritin, added to synthesizing iron-containing proteins, or secreted out through ferroportin into the circulation for recycling. Right? So a lot of iron that enters the plasma in our blood. We have about 4 mg coming from our dietary intake. About 20 mg is coming from degradation of existing red blood cells released into plasma, and then this is reincorporated into reforming red blood cells, right? Which is a really active process. Okay. And then that heme, recall that porphyrin structure can then be further degraded, bilirubin, and then conjugated to albumin for transport to the liver for further metabolism and excretion in our bile. Right? So if we just kind of trace the erythrocyte life cycle, we have it being born here in the bone marrow. So heme, iron, and globin. This is positively regulated by the circulating cytokine released by the kidney, erythropoietin. It's in circulation for 120 days. It's recognized as an old blood cell. It's phagocytosed by a macrophage, broken down, releasing amino acids, iron, and heme, which is further broken down, right? And then that iron can be reutilized, so it would come out through ferroportin in the ferrous state, modified back to ferric iron, conjugated to transferrin. Maybe it's circulated back to erythrocytes or to the liver for storage in ferritin, right? So it has a few different potential pathways. And that bilirubin can be transferred to the liver for excretion in bile. Okay. Any questions on that? Okay. So we'll move on to discussing anemia and its connections with iron and some other nutrients. We'll come back to connections with nutrients as we discuss additional micronutrients and vitamins in subsequent lectures. A lot of these have effects on blood. So anemia is a general term referring to lack of sufficient healthy red blood cells or lack of enough hemoglobin. So this is going to reduce our ability to move oxygen to our tissues in our body. And this is a really general term. There are a lot of different ways that you could go about having anemia, right? Iron deficiency anemia is one of the most common types. If you lack iron, not be able to produce enough hemoglobin, this is going to result in anemia. Pernicious anemia is another type, and we'll come back to this in the next lecture when we discuss vitamin B12. So hematocrit is a way of measuring the concentration of blood cells in our blood, so red blood cells in our blood. And basically this is just, you take blood, you spin it in this microhematocrit tube, there's centrifugation, and then you can quantify the amount of packed red blood cells as a volume of the blood. So this is just measuring the amount of erythrocytes in the blood. So about 40%, 5% is about right. So here you can see an actual This is an illustration. Here's an actual microhematocrit glass capillary tube, and this is packed hemoglobin present at the bottom. This is going to be plasma, and then this interface, which appears white, it's what they call the Buffy coat layer, is mainly the white blood cells. So for something here where we have a normal distribution, about 45%, if you did this test and you had something like this, this would indicate some sort of anemia. Right? This could be due to iron deficiency or some other cause. Okay, so the hematocrit's a useful way to measure red blood cell content in the blood and determine if there's actual anemia. But also, once you determine this, it's useful to look at the morphology of the blood cells because there are different types of anemia which will be indicated by the different type morphologies of the blood cell. So erythroblasts divide and mature within the bone marrow, as I've discussed. And during this time, heme and globin are synthesized, stored as hemoglobin in these dividing cells. And this is the important part. As division proceeds, the cell size becomes smaller. Okay? And normally the cell division stops when hemoglobin concentration reaches about 20%. So this is a pretty high concentration of hemoglobin in these cells. But if hemoglobin is limited, for example, if you have iron deficiency, the cells will divide more times than usual. So you're going to start to get cell size that's smaller than usual. And this is referred to as microcytic anemia, in this small cell anemia. You'll get smaller red blood cells than normal under conditions of iron deficiency. And this is due to this lack of hemoglobin. So the cells will abnormally keep dividing. And you'll get these tinier red blood cells. So this iron deficiency anemia develops during long periods of insufficient iron intake. This is opposed to something like a rapid anemic state, where you just lose a lot of blood cells. You'll have low iron, but you won't develop immediately this microcytic anemia. So early on, iron stores within our body are going to be able to compensate for low intake of iron. So if you start consuming diet that's low in iron, we have quite a bit of stores in our body that can compensate for this. Release from ferritin stores in our liver and transport, so we'll continue to produce hemoglobin and continue to produce normal functioning red blood cells. In addition, those red blood cells can live for 120 days. They can live for a while. So there's a good amount of compensation for inadequate hemoglobin and inadequate iron. But if iron deficiency continues, eventually the cell size will stay small and the percent of hemoglobin will continue and will develop this microcytic anemia. So this can occur, I've talked about iron, but this can occur with deficiency in a lot of different nutrients, this microcytic or small cell anemia. So iron, if we don't have enough iron, which is necessary, it's incorporated into heme, it's necessary for the production of heme and hemoglobin and productive functional red blood cells, we'll get this microcytic anemia. If you don't have zinc, so remember this is a cofactor right, for the ALA dehydratase, one of the enzymes involved in heme biosynthesis. You'll also get microcytic anemia. PLP, that vitamin B6, the cofactor for ALA synthase, also give you microcytic anemia. Copper is actually required for ceruloplasmin, which is the enzyme that oxidizes ferrous iron to ferric iron, facilitates binding to transferrin. So you'll also get reduced delivery of iron to tissues and reduced hemoglobin production in this microcytic anemia. So any of these can cause microcytic anemia. Same with lead toxicity. So lead does a lot of things, and there's a lot of reasons it's toxic. But one of the things that it does is it results in microcytic anemia by blocking multiple steps in that heme synthesis pathway, right? ADA dehydratase and the ferrochelatase are both inhibited by iron. So a lot of these factors can result in this microcytic anemia. Okay, so I think we've reached the end. Are there any questions about today's lecture? Question?
Perhaps is the reason why the erythrocytes keep dividing in microcytic anemia. Is that because you don't have enough iron and then your body doesn't produce as many cells, and then so they're like, oh, let's keep dividing
So the ego